Culturomics is mainly a biochemical approach to identify strains or species in a complex sample, like that from the human gut or a microbial environment. This involves the manual labor of culturing microbes in the wet lab and then follow it up with the identification technique such as MALDI-TOF. Important to note, this is not a complete alternative to metagenomics, but rather a complementary approach to highlight some difficult to detect species. From a metagenomic standpoint, culturomics is a rather powerful technique to identify bacteria that could easily classify as a dark matter if their phylogeny or sequence information was not already known. This means that a new strain or species could be identified with culturomics and then followed up with whole genome sequencing to properly profile its taxonomy. Since metagenomics techniques like 16S or shotgun sequencing use homologous reference sequences to identify the genomes in the sample, they are more prone to identifying DNA signatures from dead microbes, or in other words, functionally inactive DNA. With culturomics, we bypass this limitation because only the living bacteria is selected in the first phase of culturing. Another advantage here is that with this culturing, we can also capture low abundance species or slow growing bacteria so that their presence is always known. Recent research in this field has shown that increasing the number of samples is a favorable strategy for capturing overall microbial species diversity, which indicates that some bacteria can evade detection if we only rely on one sample to represent the environment. And that makes sense as there could be variation in results due to individual differences in samples. So, how do we detect those species? The most popular approach in the market is the MALDI-TOF mass spectrometry. As a quick refresher, MALDI-TOF stands for Matrix Assisted Laser Desorption Ionization Time of Flight and essentially shoots a laser at the sample of interest and ionizes its biomolecules. This includes proteins that basically float onwards towards the detector. This time of flight is recorded for every biomolecule and its mass is calculated in the sample. The resulting mass spectrum acts as a fingerprint for the identification of a microbe. This is so exact that using Maldito from a system like Broker Biotyper, we can detect microorganisms with an identity score. This score has a known interpretation such that only a score of more than 2 is considered a confident species detection, while anything lower could be confident for genus level or even unreliable. Another interesting approach is using machine learning on the cultured microbes. Recently published CAMI robot, which stands for Culturomics by Automated Microbiome Imaging and Isolation, uses computer vision to leverage colony morphology. This is basically the visual appearance of the colony and the genomic data to identify the strains in the culture. Essentially, the colony size and shape are extracted from illuminated images, following which the algorithm picks colonies that appear different from one another. This allows strain prediction and identification. Furthermore, this approach could be mixed with mass spectrometry in the future and then become even more precise in identification. So, while a culture independent approach like 16S sequencing cannot see not yet cultured species or even those low abundance bacteria, the culture dependent approach like culturomics is limited with information on only the culturable bacteria. Bacteria that require specific cultivation conditions or those that cannot be recovered with cultivation conditions may be missed. Therefore, both approaches, culturomics and metagenomics, are complementary to each other, and their strengths can be leveraged to gain a more comprehensive understanding of the gut microbiome or a complex microbiome.